Hello students, in today's video we are going to discuss about the early development of embryo and also implantation. Before we study the implantation or early development of embryo, let us first understand the events which are taking place before fertilization. Already we have discussed in our last video, but one more time I want to recall all the events which are taking place before fertilization. So, fertilization, at the time of fertilization, egg should be there in the fallopian tube. And this egg release takes place on the 14th day of menstrual cycle. That process we call as a ovulation. And at the same time, if the male inseminate the sperms and egg has to reach the region of fallopian tube, that is ampullary ischemic region. And when both the sperms and egg reach the same time, this ampullary ischemic region, then only there is a possibility of fertilization. Otherwise, the ovum is not going to get fertilized. Hence, we may see that all copulations are not leading to the fertilization, because when the sperms are inseminated, at that time, if the sperm are not able to reach the ovum, the sperms are going to be wasted. So these two, sperms and egg, reach the ampulla ischemic region, the fertilization is over and the results the single cell zygote. And early development or embryogenesis starts from zygote. So zygote is the initial cell from which embryogenesis starts. So what is embryogenesis? It's a process of development of embryo from fertilized ovum or zygote. First, we will discuss the structure of zygote at the time of fertilization, how it looks and what are the structures which are retaining during embryo development and what are losing. First of all, we will study the structure of zygote. So that zygote is single cell diploid cell which has diploid nucleus. Nucleus is here. This is the nucleus which has two poles. One is animal pole and the one is vegetal pole. And this zygote has surrounded by all the three layers which were there in the egg. All those three layers will be here. This layer is called the plasma membrane or vitelline membrane, and it's surrounded by the next layer. So this layer is called the zona pellucida, and zona pellucida is externally covered with one more layer, and that layer we call the corona radiating layer, which is made by follicular cells. So all these three types of layers will be there in the zygote. So outermost layer is corona radiata, and next one is zona pellucida, and inner one is plasma membrane. Inner one is plasma membrane. So inside the cell, cytoplasma is there, all the cell organelles are there, and along with that a diploid nucleus is there. So the first division starts in the zygote after 30 hours of fertilization. So these cells divisions are called the cleavage. So zygote undergo repeated mitotic cell divisions. By that what happens? There is an increase in the number of cells, but there is no addition of the volume. So the size of the zygote or the size of the embryo will be same, but the number of cells are increasing. So this is very important. We may come across many questions in the NEET also, where in one question paper I have seen where the question is that there are 4 options. What are the changes which are taking place during the early development? So the number of cells are increasing, but the size of the embryo will be same. There is no change in the size. So whether increasing or decreasing, or both the number and size is increasing, both the number and size is decreasing, there are many such options will be there. So students, please keep it in mind. The cell's number is increasing, but the size of the cell is decreasing, because cell division is continuously takes place within that cytoplasm, and by that, for every cell division, the size of the cell is going to be decreasing, but the number of cells are increasing. So this is very important. The first cleavage starts. So after 30 hours of fertilization. After 30 hours of fertilization, the first cleavage takes place, or first cell division takes place, which takes place from the animal pole to the vegetal pole. So from animal pole to the vegetal pole, the zygote is going to be divided. So this division is called the holoblastic. This division is called the holoblastic, and this division results the two equal sized daughter cells. And these daughter cells are called the blastomeres. Now they are called as a blastomeres. So why it is called a holoblastic? What is the meaning of holoblastic? Holoblastic means the one in which the cytoplasm of the cell is completely dividing into two equal parts. Here the cytoplasm of this cell is going to be divided completely into two equal parts. Such cytoplasmic division is called the holoblastic. So human egg is dividing by holoblastic method, because in human egg the yolk is very in negligible amount. But in case of frog egg, the yolk content is very high and cytoplasm is not going to divide the into two equal parts, or cytoplasm content is not equally dividing in such situation. So, but in human being, the cytoplasmic content is equally dividing into two identical blastomeres. Hence, the cell division in human zygote is called the holoblast. And the two equal size blastomeres are forming at the end of first cleavage. So here we can see all the events which we have studied now. So this is the egg which is released from the ovary on the 14th day of menstrual cycle. That process is called the ovulation. The released egg is collected from the embryo, and it is entered deeper portion of the fallopian tube, where it will reach the or gets fused with the one of the sperm inseminated by the copulation, and the one of the sperm, when fused with the egg nucleus, it's going to get fertilized, and the fertilization process results the zygote. So zygote is a single cell diploid, the initial cell of the embryogenesis. From the single cell zygote, the multi-celled embryo formation takes place. So now we have studied up to here. So as the cleavage starts or cell division starts in the zygote, at the same time this early embryo moving towards the deeper part of the fallopian tube towards the uterus. So simultaneously this egg is going, the fertilized egg or zygote is moving. So the early embryo is moving towards the uterus. So now two blastomeres are there, and after 40 hours of fertilization, so the second cleavage takes place, or second mitotic division takes place, and that results the 4 equal size cells, and they are called the 4 blastomeres, and the stage is called the 4 cell stage. And at the end of third day, at the end of the third day, so the early embryo enters the uterus. The early embryo enters the uterus, which has 8 to 16 cells or blastomeres. And this mass of cells or solid structure is called the morula. This is the stage 8 cell to 16 cell stage is called the morula. Either it may be 8 cell or 16 cell stage, and this is the solid mass of cells stage, and this stage is called the morula. So during morula, so the structure is completely filled with the cells. There is no hollow or any cavities there. But this and the morula enters the uterus. By the time the endometrium wall secretes the nutrient fluid. That nutrient fluid is called the uterine milk. The endometrium wall secretes the nutrient fluid, and that nutrient fluid is called the uterine milk. And this uterine milk is absorbed by the outer layer cells of morula. The outer layer of cells of morula absorbs this nutrient fluid, and by that what happens? The size of the morula slightly increase. And this is the one change which takes place in the morula. And also suddenly the morula undergoes many continuous division and forms many more cells, and it turns to the next stage that we call the blastocyst. So for the transformation of morula to the blastocyst, there are many changes are taking place. The first change is the many cells which are arranged on the peripheral layer of morula are a little bit enlarging, and they are further flattening. And these flattened cells are now act as a trophoblast or trophoectoderm of blastocyst. The outermost layer of morula, these cells are flattening, and this layer which is made by flattened cells is called the trophoblast. It's called the trophoblast or trophoectoderm. Trophoectoderm. And these trophoblast cells secretes a secretion, and because of that secretion there is a cavity is formed, and that cavity is called the blastocoel. The cavity is called the blastocoel, and it is characteristic feature of the stage blastocyst. And there are group of cells at one end of the blastocyst, and these cells are called the inner cell mass. These are called the inner cell mass. So what are the changes which have taken place while transforming the morula to the blastocyst? The outer layer of morula cells are enlarging, and they are further flattening, and these flattened cells layer is called the trophoblast or trophoectoderm, and these cells are secreting a secretion which creates that cavity, and that cavity is called a blastocoel, and this cavity differentiating trophoblast from the inner cell mass, and this inner cell mass is forming a knob-like structure, and that knob is called the embryonal knob. Embryonal knob. Here one more thing is happening, that is the zygote which has intact with the three layers, corona radiata, zona pellucida and plasma membrane. While transforming morula to the blastocyst, the corona radiating layer is degenerating. Now the blastocyst is having only the two layers. The outer one is called this layer is called the zona pellucida. Zona pellucida layer is there, but corona radiating layer is disappearing while transforming morula to the blastocyst. So there is a inner cell mass, and that we call the embryonal knob. And this embryonal knob in future is going to give rise to the complete embryo, or development of embryo takes place from this embryonal knob. But trophoblasts are not taking part in the embryo formation, but these trophoblast cells or trophoblast layer is going to help during embryogenesis in two ways. One is trophoblast cells produce the finger-like structures. They are called the chorionic villi, and they are useful to penetrate the deeper part of the endometrium, and by the way it helps in the implantation. This is the one help of trophoblast. Another use of trophoblast cell is this trophoblast layer is going to give two embryonic membranes or layers. One is amnion and another one is chorion. So these two layers are forming from the trophoblast, and these two layers are giving protection as well as also nourishment for the developing embryo. So there are two poles are creating. One is we may call the animal pole and vegetal pole. Animal pole we also call the embryonal pole, where the embryonal knob is present, and the opposite pole where the no embryonal knob is there, or opposite to the embryonal knob pole is called the vegetal pole or an embryonal pole. So two poles are created, and this blastocyst when enters the uterus, it occupies on the endometrium wall. So before it occupies on the endometrium wall, there is one more thing is very necessary, that is this blastocyst has to get rid of this zona pellucida, because zona pellucida help the blastocyst for fixation in proper place. It avoids the blastocyst attachment in the abnormal site. So once the blastocyst become ready for implantation, the zona pellucida layer is going to degenerate. And this blastocyst enters the endometrium wall. So there is a depression is creating on the endometrium wall. So this is the endometrial layer. So when blastocyst arise on the endometrial layer, there is a small depression or fit is forming. And this depression or fit is forming by the enzyme secreted by the trophoblast layer. These hydrolytic enzymes secreted by the trophoblast will dissolve the certain portion of the endometrium and makes a depression or a pit. And this blastocyst occupies this place. And here, as it landing on the endometrium wall, the cells which are present in the endometrial layer are swelling and they are become losing, and the endometrium wall is highly become vascularized and thickening, and the cells which are in contact with this embryonal mass are called the cells of Robber. These are called the cells of Robber. The trophoblast cells which are in contact with embryonal mass or inner mass cells are called the cells of Robber. And these cells, they produce the finger-like projections in the deeper portion of the endometrium wall. They produce the finger-like projections called the chorionic villi. Chorionic villi. These are helpful for penetration of the blastocyst in the inner membrane of endometrium, and also they are helpful for the absorption of nutrient material from this endometrium wall. At the same time, the endometrium wall layer or uterus tissue also interdigit between this finger-like projection. It is interdigiting between the chorionic villi, and they also produce, these cells also produce a layer around the blastocyst. And in this manner, blastocyst is going to be buried in the endometrium wall. And this one we call the implantation. So we may define the definition of implantation. Implantation is nothing but it's the attachment of blastocyst on the wall of endometrium, and which takes place on the 7th day after fertilization. So this implantation takes place on the 7th day of fertilization or after 7th day of fertilization, and the whole process of implantation is going to be completed within 2 to 3 days. Means The period of implantation is 6th to 10th day after fertilization. And once the implantation is over, this is the first sign of pregnancy. And pregnancy is the period from conception to the birth of a child. In human being, the period of pregnancy is 9 months 7 days, more or less. So it may be variable in different mammals. For example, if you take the cat, the gestation period or pregnancy period is only 63 days. But when you talk about the elephants, it may be 653 days. So in the next class, we are going to discuss the further events of human reproduction. So there is a placenta. So when these two tissues are interdigited, there is an organic connection is creating that we call the placenta. So what is placenta? What are its functions? How it is helpful for the fetus? We'll discuss in the next class. Thank you.